Slater, and I'm a postdoc at the USDA Bee Lab, and we kind of focus on bee breeding and genetics. I got my start in North Dakota as a beekeeper, so I actually worked for a commercial beekeeper in North Dakota, and the beekeeper managed around 20,000 colonies. So I've really been around bees my entire life. And one thing I noticed, just kind of working for this commercial beekeeper was, this kind of queen failure issue where we would buy queens every single year, and we'd probably replace probably most queens because within 6 months these queens would be failing. And so one thing that really piqued my interest was, why are these queens failing, and can we try to understand what's the cause of this? And so from there, I decided I went to North Dakota State University, where I focused on nutrition and queen quality. I worked for the Bee Informed Partnership for a couple years, where I kind of a tech transfer program in the United States, where I sampled beekeepers from across the United States, and then I started my PhD at Purdue. And at Purdue, I really focused on drones because drones are kind of this forgotten caste. When I ask beekeepers, when you think about drones, what do you think of? They just think of, they respond. They say that drones are kind of useless males in the colonies. Their only role is a mate. They don't really do much, but they're really important, and they're really important for colony health overall. And so I really hope by the end of the talk that you realize that drones are not only important, but they're vital for any type of breeding and selection program. So first, I always like to begin because I never know sort of the level of the beekeepers in the group. And so first we'll talk about, the social structure of colony. And so colony typically has, 5,000 to 20,000 colony members. And there's around, 10,000 of them are workers, and these workers are non-reproductive females, and they perform a wide range of tasks such as guarding, cleaning, nesting, and a lot of other different behaviors. Then there is a queen, and this queen is the reproductor of the colony. She lays around 2,000 eggs per day, and her only goal is to reproduce. And then we have these males. These males are large, they're about the same size as queens, and you see they have these big eyes that help them navigate during these flights. And the males, they don't really contribute too much directly to the colony, but indirectly they contribute not only genetics, but also sperm and other aspects of sperm to the queen. And so while we may not think of drones being important, they're actually really important to not only queen quality, but overall colony health. And so one reason I really got interested with drones was, well, first of all, we don't know much about drones at all. It's kind of a forgotten field in a lot of ways. They're really the forgotten caste because, most people research that focus on the queen aspects of failure and other colony phenotypes. But, here I really decided to focus on drones during my PhD at Purdue University. And so one reason I decided to focus on drones was there's this issue of queen failure where beekeepers are buying queens and they're failing within 6 months, where queens should be thriving and reproducing for 2, 3, 4 years. And a lot of research has really been invested into the queen aspect of failure. I know Dr. McAfee at BC, I think she's at BC. She studied a lot with heat stress and queen failure. And so she's been showing a lot of research showing that there's some aspects of environmental and genetics that can cause queens failing. But for me, I really decided to focus on drones because we're starting to realize that, well, these queen failures actually may be linked to poor drone quality and poor sperm quality altogether. When we collect queens that are failing, we see that not only are they storing poor quality sperm, but less sperm than they expected. And so I really decided to focus my research on trying to identify what causes poor drones and what can we do as beekeepers to improve that. And to really illustrate this a little bit more, this is a study done by Jeffrey Pettis when he's at the USDA in Maryland. And he heard these same rumors from beekeepers where these queen failure rates are really high. And so he collected failing queens from the East Coast, the West Coast, and then newly requeened. And then he had his controlled queens at the Maryland Bee Lab. And he found that for the control, the queens are storing nearly 100% viable sperm. And so this basically means that 80 to 90% of the sperm that the queen was storing in her sperm storage organ was nearly 100% alive. And that's what we expected, and that's what beekeepers should want. Whereas queens that were failing, nearly half the sperm was dead. I mean, some of these queens, only 60% of the sperm was dead versus 80%. And so this really illustrates that these queen failures might actually be linked to poor drone quality and poor sperm quality altogether, rather than just a queen issue. Now, why should we care about drones? Now, first, I talked about queens, right? Drones are really important for queen quality overall because they not only provide sperm, but they also provide a lot of different aspects of sperm, such as proteins that can improve her longevity, it can improve her storage, and so on. But drones are also really important for a lot of these other colony level traits. And so when you think about the genetic structure of a colony, not only have the genetics come from the queen, but they also another half come from the drones. And so they're not only important for the queen aspect of some of these traits, but also these overall colony level traits. And we know that if queens mate with certain drones, that they can express some of these traits of interest. So for example, if queens mate with certain drones, they can elicit mite resistance, viral resistance, higher productivity, different landscapes, and overall colony level performance. And so drones are not only important for queens, but also some of these other important traits that you may be interested in as a beekeeper or breeder. Now, what actually makes a successful drone? And this was kind of a major question I had before I even entered my PhD. Like, when you think about a high quality and successful drone, what does that mean? And I could really sparse it down into kind of these three major points. Point number one, sperm production. Point number two, sexual maturity. And point number three, mating behavior. And this whole life cycle and these three major points occur during this kind of 54-day lifespan. Now, point number one is sperm production. So during development of a honeybee drone, this is when sperm is produced. And so drones can never produce sperm ever again. It only occurs during this 24-day period. Number two is when drones reach sexual maturity. Now, drones' sexual maturity isn't directly tied to sperm production. It's more tied to other aspects of sperm movement and migration, which I'll talk about a little bit more later. But really, when drones emerge as adults, they spend the first 5 to 10 days in the middle of the colony taking care of other workers, and this is when drones actually reach sexual maturity. And this is kind of like the teenage stage for drones. And then once drones reach sexual maturity, they go on these mating flights, and they either successfully mate or they die trying. And like I said, this whole process takes around 54 days. And so just remember that these three major points for what makes a successful drone: sperm production, sexual maturity, and mating behavior. So point number one, so honeybee drones have this holometabolous development, which means that they go from an egg stage to a larval stage to a prepupa stage, and then to kind of this full pupa stage. Now, for the gonads are actually developed during this egg stage, and they actually grow bigger and bigger during the early instar larval stage to the big larval instar stage. And so if you were looking at like a drone or like a drone frame, for example, the testes are fully developed when the drone larvae are really big, and they're really, it kind of looks like they can't really fit inside the cell anymore. And this is what we call the fifth instar larva. And the exact terms don't really matter too much, but really what really matters is, one, the testes are fully developed at this stage, and this is when sperm is actually first produced during this kind of late larval instar. Now, around a day or 2 days after that, the workers actually cap the cell, and the drone goes through this kind of cocoon spinning stage. And this is kind of known as a prepupa stage. And this is the second stage of sperm production. And so really sperm is only produced during these 2 days of development, this late larval instar to the prepupa stage. Now, drones can never produce sperm ever again. It can only occur during these 2 days of development. Now, then once drones reach pupation, where you can see the full body form, the whole reproductive tract is formed, and the eyes are kind of this pink-eyed, purple-eyed stage. This is when the sperm tails are actually developing. And so sperm is produced in these first two stages, but the actual tail, which is important for fertilization, swimming, and other quality metrics for sperm, is occurring during this stage. Now, I want you to kind of remember this. Sperm can only be produced during this stage. And so if anything happens during this stage, that can cause some long-term and really irrecoverable damage to honeybee sperm. And just think about what happens during this stage. First, a lot of mite treatments are being applied during the stage. And then second, these drones are really attractive to mites, and they're really mite magnets in a lot of ways. And so if mites are jumping into these cells during the stage when sperm is being produced, that can cause some long-term irreparable damage to drones overall. And so I won't talk about that too much at all, but just remember that this stage is extremely sensitive and really can cause a lot of damage to honeybee males' fertility. Okay, point number two is sexual maturity. So once drones emerge as adults, they spend around the first 5 to 10 days in the middle of the colony, and they really can't feed for themselves at all. They need to be taken care of by the workers, so they receive all the feed and care by the workers in these first 5 to 10 days of development. And this is extremely important because this is a stage when drones actually reach sexual maturity. Now, unlike a lot of close related taxa or a lot of organisms overall, sperm or sexual maturity for honeybee drones isn't really connected to the initiation of sperm production. It's associated with the swimming of sperm from the testes to seminal vesicles. And so the reason drones aren't sexually mature when they emerge as adults is because the sperm is actually stored in the testes and it has to swim to seminal vesicles in order to be used during these mating flights. And so that's why the drones really spend these first 5 to 10 days in the middle of the colony, because the sperm needs to swim to seminal vesicles and really mature in order for the drones to properly mate. And what's really cool about this stage too is workers can really sense where the drones are at. So drones are lower quality or they're really mature and a lot of sperm are still stored in the testes. The workers actually give them more vibrational signals, more food signals, and they give them a lot more environmental signals to really either speed up the maturity or improve quality overall. And so this is what a immature drone versus a mature drone looks like. And so when you have the chance this summer or maybe the next few months, whenever your colonies begin producing drones, if you haven't done this before, just kind of squeeze on the thorax of the drone and the endophallus, which is this clear structure, will pop out. And from there, you can kind of visually ID whether this drone is mature or immature. Now, this is what an immature drone looks like. You can see the endophallus is clear and there's no kind of white mucus protein or brown sperm at the tip of the endophallus. Whereas a mature drone, you can see that this isn't the best picture, but you can see this online if you Google it. Actually, I think with the Everbright invitation, there's a picture of the drone that was actually mature. But from that picture, you can see that there's this kind of brown spot at the tip of the endophallus, and that suggests that the sperm is mature and it's migrated and it's really ready for drones to begin mating. And so the really biggest takeaway for this point is drones have to spend these first 5 to 10 days being taken by the workers in order to reach sexual maturity. And after this, they can really begin their mating flights. And so I get to the point number 3 is kind of these mating flights. And so drones go to these areas known as drone congregation areas or DCAs. And these DCAs can have 1,000 to 25,000 males. And these DCAs are usually the same year in, year out. And so if there's a reported DCA 50 to 60 years ago, it's pretty likely or highly likely that if you go to the DCA now, that the drones will still be flying to those areas for mating, which is really interesting. And it seems like drones use kind of these visual cues and partially some pheromone cues to really locate these drone congregation areas. Now, when drones reach sexual maturity and they begin these mating flights, they'll go on what is known as sort of orientation mating flights, where they'll go to multiple drone congregation areas. And then after that, they'll kind of pick and choose one or a few, because once they get older, they have less time to really mate before they die. So they really pick one or two and go back and forth between them. And they go on these flights for around 20 to 30 minutes, which I'll illustrate a little bit more at the end of the talk. But they'll really do this until a queen arrives. And then once a queen arrives, they'll form these giant drone comets of all these drones, and they'll chase the queen. And it's a really competitive environment where drones are really competing with each other for female access and to really mate with the queen. And so I'll show that a little bit more later on in the talk. But really, what to remember here is, when you think about what makes a successful drone, but more importantly, what causes a poor drone, are these 3 major points. 1, sperm quality. 2, sexual maturity. And then 3, mating behavior. Okay, so what actually can cause a poor drone? And the first are these kind of environmental factors. And I split it down to 5 major factors, and I'm sure there are more, but these are the environmental factors I deem the most important and the ones that can really cause poor drones. And this is from a lot of research done throughout labs in the US and Canada, especially Julian Rangel's lab and some other labs in Canada, versus poor nutrition. So if we know that a colony is receiving poor nutrition, whether it's a dearth or whether or not receiving the proper diversity of nutrition, this can cause poor drones. We know certain pest pathogens, diseases can cause poor drones, such as mites, the foul brood bacterial diseases, fungal diseases, and nosema. We know certain miticides can infect drones. So chemical caricides or synthetic caricides had the largest effect on drones, and even some organic caricides such as oxalic acid and formic acid can still have a small effect on drones. So really, all miticides have some extent effect on drones. For pesticides, we know that neonicotinoids, fungicides, and other pesticides can impact drones, and then temperature, which is a lot of studies being done at BC University. And so all these different factors, they've really been well studied. We know that they can have a huge effect on drone quality, especially if they occur during this developmental stage. But today I'm going to be really focusing on the genetic causes of this variation. And to really show this a little bit more, so let's go back to your high school statistics where we have this kind of bell curve. So let's say you sample 100 random colonies and measure drone traits from these 100 random colonies. There'll be some drones producing low quality sperm, some colonies producing high quality sperm. Now, what I'm really interested is these genes that causes a drone to produce this kind of high quality sperm, because if we can identify the genes or some of these gene networks that causes poor drones, we can really pre-screen for some of these traits. So for example, we can help beekeepers pre-screen for colonies that produce high quality drones, and we can use this in a breeding selection program, or we can use it in sort of a marker assisted selection or other genomic assisted breeding programs, which I won't talk too much into, but that's really the long-term goal of this project, is to identify these genes so we can really vastly improve the breeding and selection for some of these traits, especially for drone traits that's really hard to select for. And so I know a lot of you are bee breeders or queen producers. You already have a certain amount of time devoted to breeding and selection, and they're probably mostly towards like production or viral defenses. And so adding phenotyping for drones, for example, is really expensive and time consuming. And so the goal is to make this simpler, faster, and easier for beekeepers. And so that's why I'm really interested in the genetic aspects of poor drones versus the environmental. Okay, so first we're going to focus on point number one, and that is sperm quality. And just to summarize once again, the testes for the drones are produced first during the egg stages, and then they fully form when the drones reaches kind of late larval instar. And this is when the sperm is first being produced. It's late larval instar when the drones are really big and large. And then once the cell's capped and the drones enter this prepupa or cocoon spinning stage, this is the second stage of sperm production. And so like I mentioned earlier, sperm is only produced during these 2 to 3 days of development. Now, the sperm tail is elongated and produced during this pink-eyed, purple-eyed pupa stage. And then once drones emerge as adult, all the sperm is stored in the testes, which I'm circling right here. And so these are the different points that really impact sperm quality and sperm production overall. And one question I had was, what are the genetic factors that causes high quality versus a low quality drone? And what are the genetic factors driving that variation? And so to look at that, I identified 4 distinct breeding lines from across the United States and Canada. And I'm just going to call them line 1, 2, 3, and 4. But I started with 10 total lines, and these are stocks that beekeepers either commonly use. They're part of a breeding and selection program. And I also chose them based on genetic ancestry. So which ones had higher European lineages versus Africanized lineages. So basically known genetic differences between them. And so, for example, one of these lines had around 30 to 40% Africanized genes versus the other lines had more Carniolan or Italian genetics, if that makes sense, which I won't talk about too much about that, but if you have questions later on, I might be able to specify that a little bit more. But basically, I started with 10 lines and I found the ones that were basically produced the most different drones. And that kind of sparsed it down to these 4 distinct lines. Now, from these lines, I raised what is known as a common garden experiment. And so for me as a geneticist, I really hate the environment because it makes things really complicated, but it also makes it more difficult to really identify the genes that, I really want to use for a pre-screening or a marker assisted breeding program. And so we did what is known as a common garden experiment, where we brought queens from these different lines and raised them in one environment in central Indiana. And we did that because we could control the environment to the best of our ability. So we could control for the time of the season because they're treated the same. We control for the miticides and these other environmental factors. And basically by doing that, we can really identify certain genes that causes poor drones, what causes quality drones. And so we raised drones from these 4 different lines, and then once they emerge as adults, we put them into a common colony so we can control for all the different environment. But basically, these drones are treated exactly the same environmentally. So just keep that in mind as we go forward. And then I sampled drones at ages 10, 14, and 20. And I did this because drones undergo what is known as a reproductive senescence, where sperm quality is low, it increases and maximizes at age 14, and then as drones age after around 2 weeks, sperm quality traits decrease over time. And this is because, when drones emerge as adult, the sperm is beginning to migrate, and that migration is really complete by age 14. But then after that, just due to flight stresses, environmental stresses, and just aging overall, the sperm quality decreases over time. And so that's a major reason I sampled at 10, 14, 20, so I can capture all this variation. And that measured a wide range of sperm traits. I won't name them all, but I looked at the amount of sperm they were producing, the percent viability, so what percentage of the sperm was alive that the drones produced, how fast was the sperm producing, and other morphometric measurements. But really, when we report throughout this talk is just how much sperm these drones are producing, because that was highly correlated to a lot of these other traits. And so sperm numbers are really good metric for looking at sperm traits overall. And these are my results for my 4 different lines. So this is line 1, 2, 3, and 4. And these are our 3 different ages, 10, 14, and 20. And this is the amount of sperm in million sperm cells. So for example, 2 million sperm cells, 4 million, and 6 million sperm cells. And so this is typically what the amount of sperm that a drone typically produces, around 2 to 12 million sperm cells. Now I want you to focus on these 2 green highlighted lines, line 1 and 4. Now first, you can see that they have this senescence where sperm number is really low at age 10, increases maximized at age 14, and then decreases as drones age to age 20. Then line 4 also had this senescence. But what's interesting is this amount of variation we saw between drones at line 1 and 4 at age 14, where line 1 drones are producing nearly 6 to 8 million sperm cells at age 14, versus line 4, these drones are producing 2 to 4 million sperm cells. And so that's about a 2 to 3 million sperm cell difference. And to really think about this a little bit more, these drones were raised in the exact same environment. They're raised by the exact same workers. The environmental factors were exactly the same because, they're raised in the same colony. Yet these drones are producing 2 to 3 times more sperm than this line. And this really shows that environmental factors really isn't driving poor drones for these lines. It's really genetics that's causing line 1 to produce high quality drones versus line 4 from producing low quality drones, because these drones are treated exactly the same. Now to look at how much genetics actually plays a role in line 1 drones producing 2 to 3 million more sperm cells, we looked at what's known as a heritability metric. And this metric ranges from 0 to 1. 1 means that a trait is determined nearly 100% by genetics. And so an example of that would be eye color. So we know that there's certain genes that can cause a person to have green eyes, blue eyes, or brown eyes. And typically a heritability for eye color would be nearly 100%. Whereas a heritability of 0 would mean that a trait is determined nearly entirely by the environment, and there's no genetic factors causing a trait of interest. Now we found that 33% of genetics was explaining this variation. Now that doesn't seem very high. That's actually really high for a trait such as fertility, because in a lot of other livestock, heritability for fertility traits is nearly 15%, where we saw it was nearly 33%. And so this shows that not only is we saw a lot of variation between these 2 different lines that you as beekeepers commonly use, but this genetics actually causing line 1 to produce really high quality drones. Now we didn't want to stop there. We want to identify the specific genes that causes this. And so as part of a pre-screening tool or a marker-assisted breeding tool, we want to identify the specific genes, because if we can identify the genes, we can pre-screen colonies for these genes of interest. And so what we did was we extracted testes tissue from 500 drones, and we looked at what genes are expressed in drones producing high amounts of sperm versus drones producing low amounts of sperm. And so we found around 1,200 genes that's involved with sperm production overall. And the exact methods of finding this isn't really important, but what is important is these genes are usually in what is known as sort of a gene network, where on the outskirts, there are these genes on the periphery, and these genes have a small effect. So they have effect on a trait, but they're very small effect. But then there's usually genes in the middle that have this huge effect on a trait. And so these are the genes of interest because they're not only regulating a trait, but they're regulating all these other genes involved with that trait. And so you pulled out that gene of interest because that's likely involved with, high quality versus low quality drones. And so we pulled that gene, and first of all, we actually found that this gene had significant higher expression in drones that produce high amounts of sperm versus drones that produce low amounts of sperm. So this shows that this gene appears to be involved with sperm production and high quality drones overall. Second, we found this gene was involved with what is known as dynein. And I want you to think about dynein as the motor that runs the sperm rudder. And so a sperm anatomy typically has the head, which has the DNA. We have this mid piece, which is the motor, and then we have the tail, which is a rudder. So you can think about it as sort of a boat. Now, what we basically found was the motor wasn't working. So the head was fine, the tail was fine, but the motor just wasn't working properly for these drones to produce low amounts of sperm. And so that can mean that something's wrong with the motor, whether a lack of spark plugs or a lack of gas, something's happened to the motor that's likely causing drones to produce low amounts of sperm. And so just keep that in mind. Dynein means sperm motor, because that'll be really important as I go further with my talk. Okay, so first we focus on point number one, and we found that not only was there a lot of variation, but this variation was driven by genetics. We also found that dynein, which is basically the motor for the sperm, wasn't properly working for drones producing low amounts of sperm. The next we get to point number two, and that is sexual maturity. When once drones emerge as adults, they spend, like I said earlier, the first 5 to 10 days of development in the middle of the colony. And they do this because they receive this constant care by the workers so drones can reach sexual maturity. Now, sexual maturity isn't driven by the initiation of sperm production because really that already occurred during development. Sexual maturity is tied to the sperm swimming from the testes to the seminal vesicles. And so that's one reason why drones really spend time in the middle of the colony, because they need to receive care to reach sexual maturity. And this is what a mature drone looks like versus an immature drone, where if you pop the drones, you can see that they have sperm at the tip of the endophallus. And so next, we're really interested in sexual maturity. And so to look at how much variation there was in sexual maturity traits, we did the exact same experiment design where we did this common garden experiment where we had our 4 distinct lines, raised them in central Indiana, and looked at whether drones were mature or immature. And so we raised drones in these 4 different colonies, and then once they emerged as adults, we put them into a common colony. And from there, we looked at sexual maturity at these 3 different ages, 10, 14, and 20. And we did that once again, just because we expect the senescence where maturity is supposed to increase over time, and then most of the drones should be sexually mature by age 14. And really, most of drones, 100% drones, should be sexually mature by age 20. And really, if drones haven't reached sexual maturity by at least age 20, it's possible that these drones may never reach sexual maturity. And so we really expect that, about 50% should be mature by 10, 100% should be mature by 14, and really 100% should be mature by age 20. And if not, there could be some long-term issues with these drones. And so we basically collected these drones and measured whether they were mature or immature. Now, throughout this talk, I'm going to focus on age 20 because, like I said, most of the drones should be mature by this age. And so here are our 4 different lines, line 1, 2, 3, and 4, and this was the percent of drones that were sexually mature. And we found that line 1, nearly 75% of the drones were sexually mature at age 20. Line 2 and 3 was around 50%, but line 4 was around 33 to 40%. Now, remember this from my earlier slides, where line 1, these drones produced the most sperm, whereas lines 4 produced the least amount of sperm. Now, we also found that line 1 drones have the highest proportion of sexual maturity versus line 4. And so they could be directly tied to each other. Now, once again, what's interesting here is these drones were treated exactly the same, right? These drones were raised in the exact same environment during the same season, raised by the same workers, yet drones from line 1 have a higher sexual maturity percentage versus line 4. And so there seems to be something happening genetically that's causing this. And so, and I want to make one more note here is, only there's about 70% of these drones that weren't sexually mature for line 4. And this is concerning because, like I mentioned, drones should be sexually mature by age 20. And this means that there could be some long-term issues with these drones. And I'm not quite sure what to think about these drones, whether they're actually mating, if they're going on these mating flights. I'm not quite sure, because really by age 20 they should be sexually mature and should be flying. And so this is some question I'm going to be taking up the next couple summers to really figure out not only why these drones not mature, but also what's the long-term effect of these immature drones if they are flying and if they are mating. So next we wanted to figure out, okay, it appears that there's genetic factors driving line for drones to produce the least sexually mature drones. And so I don't have the metric here, but the heritability was around 40%, which was, again, really high. Okay, so next we want to see, okay, we saw that there's a lot of variation in sexual maturity. We see that this variation is driven by genetics. The next question is, what genes are causing this variation? And so once again, we pulled out brain tissue, because what was interesting is we found that expression in the brain was associated with sexual maturity rather than expression in the gonads. And so this means that sexual maturity is more tied to maybe behavioral changes in the brain. And this kind of makes sense, because in order for drones to initiate their mating flights, and drones actually had to go through these behavioral changes in the brain in order to begin that. And so that sort of makes sense that sexual maturity should be more tied to the brain rather than the testes and the gonads. And so we found that there's 825 genes associated with sexual maturity in the brain. And like I said, there's genes on the periphery that have an effect on sexual maturity, but they have a small effect. But there's usually genes in the middle that have a huge effect on a trait. And so once again, we pulled out the gene to see, what is this gene doing and what is it? As we pulled out the gene, we found that this gene was known as RAS32. And this RAS32, you can think about it as sort of the truck driver or the UPS driver. And this UPS driver basically loads up cargo, nutrition, and all these things that sperm needs, and transports it from the brain and the thorax all the way down to the testes, and actually drives these nutrients and this cargo directly to dynein, the sperm motor protein. And so this basically means that sexual maturity in the brain is directly tied to this dynein through the transportation of these nutrients, for example. And if you look at the expression between mature versus immature drones, we see that drones that were mature were producing really more of these UPS drivers. Now, what does this mean? Earlier we saw there's a lot of variation on not only sperm production, but also sexual maturity. And that can mean that something's happening to this UPS driver where either they're running out of gas, there's not enough UPS drivers, or somebody's popping their tire where the UPS drivers aren't able to get to this sperm. Either way, something's happening, probably the UPS driver or the sperm level, that's causing low sexual maturity or low sperm quality traits overall. And so something's happening at one of these stages that's causing poor drones. Now, we didn't want to stop there. So we know that the UPS driver is being expressed in the brain, and we know that the sperm motor that drives the rudder is being produced in the testes. Now the question is, how does that UPS driver actually get to the testes to actually help the sperm? And so that was our question, because we had to get from the brain all the way to the gonads. And so we pulled out those genes to see what were they doing and how were these two processes involved. And you don't know about the exact methods, but what we found was really this gene is known as BCAATD, and it's really the highway that transports cargo. And so what this does, it's a protein that really takes the UPS driver, moves it across the bloodstream of the drone all the way down to the gonads to bring those nutrients directly to the sperm. Now, you don't have to know exactly about the genes or the mechanisms or how they're involved, but the really important takeaway here is that we're seeing poor drones, and they're driven by these three genes, and there's something happening at one of these three levels that's causing poor drones, whether it's happening at the UPS driver level, such as the lack of gas, somebody's popping their tire, or there's not enough UPS drivers. Maybe something's happening at this highway level where there's road construction, or the highway just isn't built, or something at the sperm motor protein where everything is working functionally, except the motor just isn't working, whether it doesn't have gas or something. So we're seeing poor drones, and something's really happening at these three different levels that's causing poor drones. But what's really cool here is that these three genes appear to be involved with high quality drones. And so we can use this as either a pre-screening tool or actually a selection tool for a beekeeper or a bee breeder to select for, drone traits. Oh, okay. And so we have these three major points, right? Point number one, sperm quality. Point number two, sexual maturity. And then point number three is mating behavior. And so all these three traits combined really impact the sperm quality or successful drone. Okay, so next we really focus on sperm traits and what are the genetic factors causing poor versus high quality drones. Next is mating behavior. And so once drones reach sex maturity, they go on these multiple mating flights and they go to these areas known as drone congregation areas. And really when they reach sex maturity, they go to what is known as orientation flights, where they go on to multiple congregation areas and they choose these based on visual cues. And they're usually in these kind of valley areas next to either a mountainous area or next to a forest. Actually, a lot of them are found near like football stadiums because it's really flat in front of a football stadium, and then the structure gets really large. And so it appears that drones kind of use this kind of flat to upright structure to really locate these drone congregation areas. And so if you really are interested in finding these areas, maybe go to a football stadium, look up a virgin queen, or collect some drones and see if you can find them. But usually these congregation areas have between 1,000 to 25,000 males, and basically drones just congregate there basically to wait for a queen to arrive. Okay, I want to show the video quick of a comet. So this is a queen that's about to go on her mating flight, and so you see the queen beginning her mating flight. And you can kind of see this cloud of drones right here, and they're chasing the queen. Now this queen's flying, and these drones are competing with each other for female access. You see, it's a really competitive environment. These drones are knocking each other out of the way. They're really fighting with each other. They want to successfully mate the queen. Now this drone actually latches onto queen and begins mating. And once the drone actually inseminates the queen, the drone just becomes paralyzed. The wings go up and the drone's mating, and then they actually do this free fall. Then once mating's over, the drone actually dies and falls to the ground. And so the drones can only mate in their lifespan. And then once this mating is over, the queens will go on more mating flights and mate with around 16 to 20 males. That was quick. Yeah, I might start my presentation up again, but load that up. Okay, so drones only mate once, and so it's really important for them to not only find a DCA but locate a queen at these drone congregation areas. And to really show what the geography of these DCAs look like, this was a really cool study in Puerto Rico. This was actually the study that found that these DCAs are usually near these flat areas next to kind of a high rising area, whether that's a forest edge or within a valley. And so these triangles are these different apiaries. And you can see there's about 23 apiaries in this one location. And these squares are these drone congregation areas. And they found these congregation areas by putting these tracking devices on their drones. And they really found that even though there's around 20 to 30 apiaries in this location, there's only about 1, 2, 3, 4, about 5 to 6 drone congregation areas that the drones are going to. And so once drones reach sexual maturity, they'll go to 3 to 4 of these congregation areas. And then as they age and mating becomes even more urgent, they'll kind of pick and choose 1 or 2 of these DCAs and go back and forth between them. And these mating flights take around 20 to 30 minutes, and they get longer as the drones age with time. But they're also really environmental dependent, right? So if it's raining, drones aren't flying. If it's really hot, drones will change their mating behavior based on some of these environmental factors. Okay, so next question was how much does these flight traits vary? And so we looked at sperm traits. Next is looking at flight traits. Now, instead of using this fancy device to see exactly where drones are going for, this tool is really It's really useful because we can see exactly where the drones are going, but it's really time consuming and it takes a long time to really locate these drones. And it's really not very high throughput. So we can only do this on a few drones versus, I was interested in thousands of drones because I want to connect flight to some of these sperm traits. And so instead, I opted to do what's known as the RFID tag. And these are these small tags that you put in the back of the drones, and they're not GPS trackers. So we don't know exactly where the drones are going, but it gives us really useful information on the coming and goings of drones. So we can see how often they fly, when they first fly, how many flights per day are they taking, how long are these flights, and really some really important flight trait data that we can hopefully connect to some of this mating behavior data. And this is what a single drone tracking looks like. So you can see for the first 3 to 4 days, the drone's within the colony and not flying at all. Then on day 5 or 6, this drone began flying for around 20 minutes per day, got up to around 35 minutes per day. And then right here, you can see that this day it was actually raining. And so the drone wasn't flying that day. And then after the next day, the drone began flying. And so, it doesn't tell us exactly where the drones are going, but it does give us a lot of cool information about flights. And so from these 4 different lines, I marked around 2,000 drones with these RFID tags and put them into one common colony. So basically, they're surrounded by the same workers, the same environmental factors, and really everything was exactly the same. And I also isolated this away so these drones wouldn't drift to other colonies. And I really measured some of these flight traits. And this is what we found. So we first looked at age of first flight. So when did drones actually begin their first flight, which we could maybe associate with when they actually begin their mating flights. Now, I don't have line 4 here. I don't have it on here for some reason, but for line 1, we found that they begin their flights around age 8, whereas line 2, 3, and 4 was around the exact same, started their flight around day 6. Now, while 2 days doesn't seem like a lot, for a left-footed drone, 2 days is massive. And so drones from line 1 are actually delaying flight by 2 extra days. And so that can mean a few different things. They're delaying flight to not only receive more care by the workers, but they're also delaying flight to improve fertility, improve quality overall. And one other thing to note here, line 1 drones also produce the highest quality sperm. And so it's possible that they produce the highest quality sperm and high quality drones because they were delaying flight by an additional 2 days. That's an extra study. That's a hypothesis I have, but we haven't tested that yet. But it's possible that by delaying flight, that could actually improve some of these sperm traits. Next, we looked at flights per day. So line 1, they took around 10 flights per day. Line 2, 8 flights per day. And then line 3 and 4 was 6 flights per day. And so, so line 1 took around an extra 4 to 6 flights more than line 3. And so that was a lot of variation in some of these flight traits. And we also looked at, how long they were flying, and line 1 drones were actually flying for a longer distance. So not only was line 1 produce the drones with the highest quality drones, but they also produced drones that delayed their first flight and flew the most. Okay, so the next question is, okay, so we found that there was a high proportion of these drones that didn't reach sexual maturity by age 20. And then the question is, what are these drones doing? Are they staying in the colony, or are they actually going on some of these flights? And so our next comparison was comparing mature drones versus immature drones. So we first looked at first flight age. So when did drones actually begin flying? And so mature drones, they began flying around age 6, and immature drones was 6.2. And so while this is significant, I think the biggest takeaway here is that these immature drones are beginning their first flight at around age 6. And so even though some of these drones didn't reach maturity at age 20, they are initiating these flights. Next, we looked at flight duration. So mature drones flew for around 27 minutes per day, whereas immature drones flew for around 30 minutes per day. So once again, this was significant, but the biggest takeaway here is that these immature drones are flying. And so the question is, what's happening during these mating flights if they are actually mating with the queen? And so to kind of summarize, we saw there's significant variation in these 3 major points. We see that for point 1, 2, and 3, that there's a lot of variation in these 4 lines that you as beekeepers commonly use in sperm production, sexual maturity, and mating behavior. We see that some of this was driven by genetic differences, and we actually found specific genes that were causing this. And so this is really important for breeding and selection, especially if you want to start to pre-screen colonies for some of these traits. So what does this mean? There's some considerations. First, poor drone quality may contribute to queen failure. And so at the beginning of talk, I was really focused on this queen failure issue where beekeepers are buying queens and they're failing at a really high rate. The next question is, are poor drones actually contributing to this queen failure? So we see that line 1 drones line 1 is producing the highest quality drones, whereas line 4 is producing the lowest quality drones. What happens if we inseminate queens with line 4 versus 1? Will the queen perform better or worse? Will the queen fail faster? Will the queen lay less eggs? These are all questions I have, but really the major question I have is, how does poor drone quality contribute to queen failure? The next thing is performance may differ based on location. And so while these results may seem really concerning, I want you to take a step back and just take it with a grain of salt, because this is a single common garden experiment done in one location in central Indiana. And so, for example, these line 1 drones may perform better in Indiana versus line 4. But if we repeat this experiment in, let's say, California, in Florida, Texas, we may actually make it the exact opposite results, where line 4 may perform better than line 1. And so while we can't always reduce everything down to genetics, there's also still environmental things we aren't considering. And so even though it seems like the line 4 drones are the worst, if we repeat the study in their selection location, those drones actually may perform a lot better. And so really just take this with a grain of salt. Next thing is, breeding for production may actually impact fertility. So the question is, if this is truly driven by genetics and there's these genetic differences between lines, what's actually driving that? And it's possible that beekeepers are breeding for production and that's impacting fertility. And this is done in a lot of other livestock, where if livestock are breeding for milk production or other production traits, that has a negative effect on male fertility and female fertility. And so, if beekeepers are really focused on breeding for yield and viral defenses, by breeding for these traits, even though they're really important for your industry, colony health, and survival, this may have a negative impact on some of these fertility traits. Now, the question as the industry is, these traits are really important to select for, right? Viral defense and yield are really important for your business and your livelihood. And so are you willing to take a hit for fertility or male fertility? Maybe, because viral defense behaviors are really important for health and survival. And so, one thing I really want to do is try to breed for both of them. But as beekeepers in the short term, just trying to figure out where you want to invest your time and energy. And so with that, I want to thank Brock Harper at Purdue University, Crispin, who is the apiary manager, and everybody else who helped me do my project. And if you have more questions, you can email me at my Gmail. And I also want to thank all my funding partners, APF, Project APSM, Foundation for the Preservation of Honeybees, and most importantly, the North Dakota Department of Agriculture. And with that, they have time for some questions, but yeah, really appreciate you listening to my talk, and I'll take questions from here.
Garett, can you hear me?
Thank you very much, Garett. That was awesome. And we've got a couple of people of our committee that will help take the questions and relay them to you. So far, we haven't seen a lot of questions listed on the this chat, but people are also welcome to raise their hand, and we'll try to see them and let them ask their own questions. So we can do it both ways.
Sure. There's some questions on, I'm just looking through this, on drone layers. And so drone layers, yeah, so these drones are actually, they do produce viable sperm, but they also produce less sperm, and the sperm is lower quality than drones from a typical colony. I guess I can go to the top, if that's best.
Garett, there was a question really early on that had to do with the new AFB vaccine, and wondering, so do you think that the drone sperm development capability could be compromised by the new AFB vaccine, and that the queen will be passing on to the larva during the egg laying process?
Yeah. So I mean, it's tough to say because it's such a new vaccine, and I'm not sure if they've done any preliminary study with sperm, but it's possible it might compromise it. It's tough to say. Yeah, it depends on how it's passed on, how it's interacting with the egg stage. But I believe that's how the vaccine actually works and how that's going to impact kind of sperm production. So if a queen is vaccinated, it's possible that may impact sperm production, but I can't say. I know enough about the vaccine to really say. But that's something to just really consider as we go forward with this.
And then Randy, it looked like you had a question.
Yeah, can you hear me?
Yep, I can hear you.
Okay. Hey, so the line number 4, which the drones perform poorly on. Well, somebody's producing those with a commercial operation that is going year after year. Did you interview them to find out if they have a higher rate of queen problems or than the other, say, line number 1?
Yeah, that's actually a really good question, Randy. And I didn't talk to them about. I didn't talk about the queen failures, but they were commercial queen producers, and so I don't know if they're actually tracking queen failure or I guess I talked to the beekeepers that are buying those queens and seeing if they have a higher queen failure rate. But these queen failure rates can change year in year out, and so it's tough to say it's from 1 year. But it would be interesting to talk to the actual beekeepers and
I mean, because this is a question we had a California queen producers meeting a couple of weeks ago.
Okay.
And this was a question that came up when people are talking about queen failure. To me, I say, these guys, they sell you exactly the same genetics as they use in their own operation.
Yep.
And they're not having these problems that you guys are having. So, they couldn't be, if they're going to be a successful queen producer, they have to have good bees. They have to have productive bees, and they have to, if they weren't, they wouldn't be breeding from them. So it's surprising to me that there would be I'm not asking you to name who it was, but saying that there was a common commercial line that looks like it's very poor performance of the drones that doesn't make sense. That'd be worthwhile looking into.
It would be, yeah. And like I said, this was done in central Indiana. So if I repeat this study in California, that line might produce a lot better. And so these queen producers, they may be selecting for high quality drones in their region, but if they're transporting these to different locations, they may just perform poorly, right? And so, yeah, those things to really consider is talking to them and seeing how these queens are performing in these different locations.
And another question kind of along the lines of the previous question is, does mite medication cause drones to be infertile? And I think you kind of touched on that, but maybe you can expand a little more.
Yeah, so there's been several studies looking at miticide, specific acaricide treatments on drone fertility overall. And we know that, let's see, synthetic acaricides such as amitraz has the largest effect. And so we know it can decrease sperm viability by up to 70 to 80%. We know that organic miticides can still have an impact on sperm production. And even to a small extent, oxalic acid can impact drone fertility, but to really a lesser extent. But all these miticide treatments can impact drone, especially if they're occurring during this developmental stage. And so if you are worried about mite treatment and drone fertility, I would try to stay away from some of these synthetic acaricides, especially during the mating season. And so oxalic acid or formic acid might be your best bet. But there's a really good review by Juliana Rangel a couple of years ago where she reviewed some miticide treatments on drones. And so if you want to look up her name and look up her study, she has a really good review on it.
Perfect, thanks. And Liz Huckster, looks like you have a question.
Yep. So when you looked at different lines, was one of the considerations whether feral colonies were going to be observed or Russian lines? I know that there's a fair bit of data that says that feral colonies have longer lived, harder working bees in some cases in Ohio, things like that, and whether that was a concern or anything that you might want to be looking at in the future.
Yeah, so we didn't start with feral lines, and that's something that we're doing at the lab. We're looking at kind of the genetic population of some of these feral populations versus commercial or managed populations. We didn't collect feral populations, because in order to do this drone experiment, we'd have to raise a lot of drones. And so it just makes it more complicated if we're trying to do that in a feral population. But I think it would be interesting because they appear to have some unique genetics and they could have unique traits related to drones. Now, for some of these traits, we actually started with 10 lines. And so, this site really only reported 4, but these 10 were chosen first based on genetic ancestry. So based on what percent of genetics were Italian, Carniolan, or Africanized. And so that was our first metric of choosing these stocks. Next, we looked at selected stocks. And so you did mention the Russian stock. So kind of part of a breeding program like that, like the Russian breeding program, or some of these other breeding programs that were into like Saskatchewan, for example. We chose them based on some of these breeding and selection programs. And then we chose them based on what beekeepers commonly use. So like Randy mentioned, some of these kind of commercial queen producers where they're not part of a closed breeding program, they're part of an open breeding program, but these are lines that beekeepers commonly use. And so we did start with 10 of these, using these different metrics. And then we dwindled down to 4 that had the biggest differences.
Right, right. I'm wondering if in the future you'd be interested in looking at some of those Kentucky mellifera that have no Africanized genes and are very different than anything else so far in North America.
Oh, I would love to. Yeah, and that'd be a really cool next step. And so, yeah, if I can get my hands on them, I think it'd be cool to test it against this, especially, if they do have unique genetics. I mean, that's what I'm really looking for.
Cool. Thank you.
And then there's a few more questions in the chat. If you have anything to add to the organic acids and exactly the effect they have on the drones, I know you suggested Julia Rangel's paper, but there's just one more question about that. So maybe you can expand.
Yes. And so I think the one thing I first want to point out here is that you do want to treat, right? Mites are a big issue, and we know that mites probably cause more damage than any type of mite treatment on the drones. And so that's a first thing, because I know that drones, just from doing my study, it's really hard to make sure that the mites aren't getting to the drones. And so I did it was really hard to do that. And so first of all, these drones are mite maintenance, and these mites are transmitting viruses, and they're feeding the fat body. They're causing a lot of damage to drones. And so first I want to really stress that it is important to treat for your colonies, even if it has a small effect on drones. Now, as for specific mite treatments, I mean, the organic mite treatments are going to have the least effect on a drone. And so oxalic acid, for example, it'll probably drop, if you're looking at sperm viability, so how viable is the sperm that the drone is producing, it may drop it by 5 to 10%, right? So going from 90% viable to maybe 80% viable. And that's not that big of a deal. Whereas, Amichaz, for example, is going to drop it maybe from 80 to 40%. And so like all these treatments have some sort of an effect on drones. But, if you are, let's say you're raising queens, and the queens are going on mating flights in the spring, and you're raising, drone producing colonies in the spring, then I might recommend maybe doing a brood break and applying like oxalic acid, for example, to treat for mites rather than, Amichaz. And Amichaz is really effective, just kind of picking and choosing when you actually apply it. The other thing too is that, your queens are mating with drones from these different locations. And so you can try your best to really control the type of treatments that you're using to help your drones, but your queens are still going to mate with drones from another beekeeper next door that's using a synthetic acaricide. And so even though you may do a really good job of, using an organic treatment to, improve your drones, just something to keep in mind too, is that your neighbor's drones could still be made with your queen.
Yeah, neighbors, darn.
Yeah.
So, Medhat, you have a question? Go ahead.
Yeah. Hi, Garett. Thank you for your presentation. Excellent. If I am a queen producer, and now we know this issue is about the drones, do you think there's some evolutionary aspects for the queen to mate with so many drones, and later on, just if she picks about 6 to 8 million sperm in her spermatheca from various one, to manage for some of these deficiencies in some of the drone qualities? The sperm quality.
So okay, just repeat your question. So your question is there any evolutionary implications for this, or maybe some evolutionary reasons that's causing this? Is that?
Yeah, from the queen side. That's why she mates with several drones.
Yeah. So it's possible. So I guess one reason that queens may mate multiply is to collect more sperm, right? So if she, queens mating with more drones, and they can collect more sperm and live longer, and so on and so forth. And so, yeah, it's possible. And we know from some studies too, is that, if queens are really stressed out, that they'll actually mate more often with more drones. Now, another question from me here is, if these immature drones or drones that aren't really high, I guess, fully mature, I guess I might say, if queens are mating with those drones, does that cause a queen to mate more often? So, for example, if these immature drones or drones that aren't quite fully mature are in these DCAs and queens are mating with them, is the queen going to mate with more drones because of that? Because these drones are producing less sperm overall. And those are some questions we'll have to start thinking about. The other thing too, from an evolutionary perspective, is if these drones aren't immature by age 20, why are they still in the population, right? That's something I've been thinking about, because if these drones are immature and they're not producing sperm, then why are those genetics being passed to the next generation? And so I'm still thinking about kind of questions on that part of it. And it might be due to just indirect selection on some of these traits. And so beekeepers are directly selecting for production or for defense behavior, then they could be indirectly selecting for, decreased fertility. And so, yeah, I'm not quite sure what's causing this, because like you said, if these drones are immature, then, those genes should be passed to the next generation. So yeah, those are some considerations I'm thinking about.
Heather
Thank you.
Heather kind of asked a question that along those lines too, is if the sperm number or quality drops off after mating flights. So if they do a mating flight, I think what she might mean is if they do a mating flight and it's not successful and they come back, does the quality and the number of sperm drop off with every?
Yeah, exactly. So your question is, if a drone doesn't mate and the drone's going on these multiple mating flights, does that impact sperm traits? And yes, it does. And so we know that sperm maximizing the quality really maximize that around age 14. And then after that decreases, and that probably decreases at a faster rate if the temperature's higher, or the drones are flying more often, or due to a lot of different things. But we do know if they're raised in, so for example, if drones are raised in a colony that's well fed and well taken care of and doesn't have miticides, then drones actually produce specific proteins that help that sperm live longer as they age. And so that's why it's really important. If drones are really stressed early on, then that aging could happen even more quickly. So that makes sense, because drones actually produce these different proteins that really help the sperm live longer and survive longer, even if they are taking these kind of multiple mating flights. But I have seen drones that are flying, from my study, I did find drones that were flying at 40 to 50. I didn't measure how much sperm they were producing, but it's probably not very much. But they still have a chance to really pass it to the next generation. So even though it is dropping off, they can still, pass the sperm to the queen.
Right. And then your thoughts on Paul. Oh, sorry, Liz Huckster, do you want to ask a question? I think you're muted. You need to unmute.
Thank you. Line 2 on the graph where you were looking at the quality queens, and we were looking at the different lines and when they matured, and we saw that line 2 was peaking at day 20, I think. And now, do you think that could be a stock or a line character that they were just peaking, and it would be longer and maybe even better because the longer time that you saw that they waited to go out, and these guys are even long. I don't know, just putting it out there.
No, I'm really glad you brought that up. Let me go back to my graphic, because that's one thing. Okay, yeah. So you can see from line 2, they're really peaking at age 20, whereas line 1 and 4 are peaking at age 14. And so I really stopped my sampling at age 20, but I wonder if I kept sampling at age 25 or 30, that they would peak at age 24 versus age 14, an extra 10 days or so. And so is that beneficial? It might be. So if drones are staying in the colony longer, aren't taking as many flights, they may have more opportunity to produce higher quality sperm, and so that might be a beneficial characteristic. Or it could be location dependent. I'm not quite sure, but yeah, it's really interesting that these drones seem to be peaking at age 20 versus 14 for line 1 and 4.
Yeah, it seems like as well. Say the Caucasian queen or drones fly higher than, say, the Italian queens when they're in flight, and so it might be one of those kind of weird things that Caucasians do or whatever.
Yeah, and then so for some of these too, like there's some interesting things I found too. You know, the time of day that they begin flying is different between these
Right.
different lines. Yeah, so all these different flight characteristics are, you know, wildly different from them, and so we're still learning about this interaction between sperm and some of these flight traits.
And so Rhonda wanted to know the thoughts on pollen quality. There seems to be a new idea that blueberry pollen is inferior, for example. And so perhaps some of these drones, I know they were all in the same hive, but perhaps were fed inferior feed compared to other drones.
Well, so from my site, for example, I mean these drones were raised the exact same, and I didn't use any type of pollen substitute. I just used what they were, you know, kind of foraging during that time of the year, and so these drones are treated exactly the same. But to your point about kind of pollen quality, we don't know much about pollen quality and drones. I mean, maybe there's maybe Julian outlined it. I don't remember her, you know, looking at it, but we do know that there's certain components of pollen that impact queen fertility, and so it's possible that some of the same characteristics or same nutrients also impact drone fertility. And so, for example, like p-coumaric acid or some of the antioxidants, we know that they have an impact on queen fertility. And so, next question is: are they impacting drone fertility overall? Are there better pollen sources? Right? Those are all great questions. I think the one thing we do know is if colonies are in a dearth and they're not provided any type of supplement, these colonies will produce lower quality drones. And so there does seem to be interaction between nutrition and drone quality. Now, specifically which pollen sources, we don't quite know, but there is an interaction between nutrition and drone quality.
Great. And so James has asked that we please comment on the importance of providing very large numbers of drones from a multitude of colonies. So
Yeah. So that's a really good point, because you know, if you are raising queens and you want them to mate with kind of stock that you're raising, it's really important to produce as many drones as you can. You know, because it's not like other livestock where you can put them into a pen and have them mate. You want to try to flood those areas the best you can. Now, I guess what does a large number of drones actually means? I don't quite know, and that's something that we want to look at. You know, how beekeepers should be flooding their areas for their drones to express their trait. But if you have a trait of interest and you're trying to do an open mating program, yeah, it's really important to provide a lot of drones, especially in the spring, especially in BC. I'm not sure when you guys begin your queen production, but if your colonies, or if there's not many drones in the areas, then your mating's gonna be poor quality overall anyways, because you know, like the timing's not right, or there's not enough drones in the area. So thinking about the timing of the drones too is also really important.
And I just had a quick question. When you were taking, so you took the eggs out of these colonies and put them all into a single colony, the drone eggs. And so were you finding that some of those, like queens, say in line one, were actually laying fewer drone eggs or more drone? Like, did you look at the proportion of eggs that were being laid as drones from those queens when you took from them initially, and would that have any impact on the quality?
Oh my gosh, yeah, that's such a great point. Well, so first some clarification. The drones were raised in the parent colonies, and then once the drones were adults and they were put into a common colony. I didn't actually move eggs. That'd be a lot of work. But
Okay, sorry.
Yeah, but one thing I did, I had 5 colonies per line, and so I had a total of like 15 to 20 colonies. But man, some queens are producing, because I had like a drone frame and I'd drop into a colony, and some queen the next day I'd come back and the whole frame would just be full of eggs. Whereas some colonies, like she may lay 100 eggs or so, even if I trap the queen on the drone frame. There's actually a few times where I trapped the queen on the drone frame and she would just refuse to lay drone eggs at all. She would only lay, fertilized worker eggs. So I got really excited that she's actually laying drone eggs. I come back and they're end up being workers. And so, yeah, there's definitely a lot of variation in not only the egg laying rate or, the queen's willingness to lay drone eggs. It's kind of odd, especially, I did this in the spring when they should be laying a lot of drone eggs, and so there was a lot of variation in that part of it. Now, how does that impact drone quality? I don't think we quite know that. There's a researcher in in Mississippi that's looking at, kind of these components that queens add to the eggs that can impact some of these traits on queens. It'd be cool to see if he looks at drone traits, for example. And so we know there's a lot of variation in egg size and what the queens provide to those eggs, and how does it impact drones. We don't quite know, but I'm guessing it probably has a huge effect.
Yeah, thanks. So lots of people saying excellent presentation, totally agree as well. And then there were one or two questions. I'm just trying to, Jenny asked, how far did you find drones could fly from their birth hive? I don't know, did your marker or the RFD tags, I don't think they check that, do they?
No, I don't know exactly where the drones are going. I just knew they were coming in, going from the colony. So I can't really say how far they were flying. And we also don't know the DCAs in our area too. I tried to find them, I couldn't find them, and so I don't really know where the drones were actually going.
I'll need my screen to share any minute now.
And so if anyone else has any questions, or if I have missed your question in the chat, please feel free to raise your hand and ask it now. Otherwise, thank you very much, Garett.
Thank you for having me.
Yeah, we'll just give people maybe a second or two here. Oh, Jeff, go ahead. Sorry, Jeff, I think you're muted. If you could.
I have a question. So let's say you're, I am unmuted now, or okay?
You're good.
If a queen mates with too many drones that have low sperm counts, will her production be less? Will she like have a lower productive life in her span at all? Is there any data on that?
Yeah. So we know if I guess queens mate with less drones, or queens mate or store less sperm overall, that like in the beginning you won't notice as much of a difference, but really within a few months or so the queen will start going down in quality overall, pretty quickly. And like the queen should be able to sense how much sperm she's storing, but it's up to say if she's gauging that based on how many drones she's actually mating with, or based on the actual sperm that she's storing. So for example, if a queen mates with 20 drones, but they're producing a lot of sperm, is that different from a queen mating with 20 drones that aren't producing any sperm? I don't know that. And so that's another consideration I'm thinking about.
Well,
Thank you again, Garett. This has just been awesome. So many questions and so many good answers.
I think there's still two more questions there. If we're
okay. Sorry to jump in there.
Bottom here.
So sorry, Susan, go ahead.
Yeah. Hi, this is Sue Kobe. One question I have is, you look at that randomness in the mating process, the drone congregate areas, but you also have a high rate of randomness in the spermatheca, or the competition within the spermatheca. So she's mating with 20 drones, and she's going to expel 90% of that sperm. So there's a lot of competition within the vigor of those drones moving or whatever. I don't know if you get high quality, poor quality, or if she sorts that out. Anita Collins did show that she will sort out dead sperm versus live sperm. But how much competition is there within the spermatheca for high quality sperm versus low quality sperm? I don't know. That's a big question to look at.
No, that's a really good question. And I guess the one thing I found was, I guess the drones are producing low quality sperm, right? Their sperm motor protein just wasn't working properly. And so if these drones actually are mating with the queen and she's storing that, I mean, the motor protein might be working right away, but is that going to be working, a year to 2 years down the road, right? Will that sperm be dying faster? How's the competition occurring inside there? So those sperm aren't able to swim as fast or aren't able to store as long. I mean, they might be less competitive against some of the higher quality sperm. And so, those are all really good questions.
And over time, you have a lot of variation there. I mean, I've been collecting large amounts of sperm since, the early 80s. And before CCD, before all the toxic environment stuff and miticides went on, when you looked at the syringe tip, that sperm would just swirl in a beautiful swirly motion. And I don't see that anymore, or rarely see that. So I don't know if that's less quality or what's going on there, but it's different.
Yeah, that's good. Yeah, you definitely have more experience than I have. I've only really done this during my PhD, but I mean, yeah, I mean, it could be due to a lot of these different environmental factors, right? Some of these miticides, we know they impact sperm, and so maybe they're swimming less vigorously than they did. I don't know if there was any say looking at like swimming speed, 10 years ago versus now, but I'm guessing it's probably changed.
Yeah, and the sperm counts seem lower. The queens don't last as long. I used to have some that would go 3-4 years, and if I get one that goes 2, I'm happy. Most of them are 18 months now.
So even if you do you still see
Yeah,
lower
with 10
longevity.
microliters of semen, and you do the sperm counts in those, and they're less. They just don't store as much as they used to.
Have you looked
It
at
seems
sperm count and sperm viability then? Have you looked at, or just just amount of sperm that
only if I had a big problem or something, but it's I mean, this is pretty anecdotal. It's just observations over time.
Yeah, it makes me wonder if the sperm's like lower quality, and then once it's actually stored, then that sperm just kind of dies quicker. Yeah.
Yeah. I think it's
Wonder.
that combination of those things. Maybe there's less swimming into the spermatheca. Maybe there's less longevity of that sperm over time. Things are just different.
Yeah, and especially from the line, the one line, for example. I mean, the sperm was swimming less vigorously, and so if you can imagine, once that sperm is actually inseminated or actually stored, that swimming is just going to decrease really quickly, or just stop within a few months or so. It's tough to say after the fact.
Yeah, and temperature seems to be a huge factor in that now too, that we know. If you overheat or chill drones, they're super sensitive.
Yeah, it's such a you have more experience than I have, but there's so many different factors involved with it. It's such a complicated issue, as you know.
Well, it seems like I got away with a lot more earlier, but they're a lot more sensitive now. So I'm just really take a lot more care with the drones and virgins, with temperature, nursery colonies. All those things are a lot more critical now than they used to be.
So when you do do you tend to inseminate with more sperm then now, just kind of accommodate for that, like 8 microliters?
Yeah, I do. I usually do 12 microliters instead of, I started off with 8 and that seemed way sufficient. And then I bumped it up to 10. Now I'm doing 12 just to trying to get them through a full season.
Yeah. Well, so one thing I want to do this summer is I want to pre-screen these drones for some of these genes, and then inseminate queens and look at queen longevity. So I might keep in touch with you about some of the
Oh, that's fascinating. Yeah, I love to help with that or whatever you
Perfect.
need.
I'll keep in touch. Yeah, since you're the II pro. So, and you probably have a lot more questions.
There's been so little attention to drones, and I really salute you for taking this on. It's fascinating.
It was hard. Yeah, working with drones was really hard, especially for the first couple of years.
Yeah,
Taking care of them, raising them. Yeah. Oh my gosh.
They're very fragile. Yeah.
Yep.
Yeah. Thanks a bunch.
Thank you, Sue, as well.
Anthony, I saw your hand go up a couple times. Did you want to ask a question?
Well, my question was a lot like Sue's question, but as I was sitting here, have you ever looked into the Cape bee at all in South Africa, and how they go about, breeding and whatnot, and how the drones affect the way they go about it?
Yeah, that's a good question. I don't know enough about the Cape bees. I'm trying to think about like their mating system, like if they mate with the same amount of drones. But I really can't say for sure. I'm not sure. Yeah, I have to look that up. I'm not sure much about their mating system at all. I know like a lot about kind of the Africanized like lineages and their differences in mating, but I'm not sure much about the Cape bee specifically.
All right, thanks.
But I'm guessing I'm guessing there is variations in these flights, right? Because kind of warmer temperature, there's some differences in those locations. So I'm guessing they probably fly at different times of the day, fly at different heights, things like that.
Yeah, they apparently the worker bees, almost like a laying worker, can start laying. And I'm just curious how, that may be affected in some way. I don't know enough about it, but I was more curiosity.
Oh, sure, sure. She means like yeah, that kind of gets to the point with like a drone layer, for example, like laying worker that produces drones. You know,
Yeah.
they can produce drones. I mean, they tend to be smaller, produce less sperm. The sperm is lower quality, and so they they can. So I'm guessing it's same with the Cape bees too. If the the workers are laying drones, then it's possible they are producing some genetics. But yeah, it's really tough to say. I'm not quite sure actually.
Well, there's a post-doc research project for you.
Yeah, exactly. more time, right? I mean, I'd love to go to South Africa.
Will there be any point where you think you'll be able to let us know, approximate, what sort of drones we should be looking at? Or is that really just something that won't be revealed?
Yeah, so like I was telling Sue, I mean, one thing we want to do is pre-screen for these colonies. And so what we want to do this summer is kind of, get a bunch of drones, pre-screen them for some of these genes, and then inseminate queens that basically with drones that have the gene or didn't have the gene, and see that impacts, some of these queen traits. And so eventually, this could be used for maybe some eye services, to really inseminate queens with higher quality drones, or eventually want to do this on a colony level where we can, pre-screen a colony and help beekeepers select for that. But that might be a little bit more down the road. And so we'll definitely keep in touch about that part.
Oh, that's great. Yeah. Thank you.
So I'll be back in 10 years.
I bet you'll be back before then.
Yeah.
But anyway, thanks again, Garrett. You've devoted a lot of time to us, and we really appreciate it.
No, thank you for having me, and all really great questions. Like a really great group of people.
Well, thank you. And now we want to thank some other people here too. Dan Mawson, who's our webpage manager, and our presentation committee for their dedication to help this series be such a success. And if you've enjoyed this talk, or you want to find out and see, or go back and look at more of, say, Garett's slides, which were all great, and need that, you can go back to our Bee Breeder site. We have a link, and within a few weeks, we hope to have his talk posted there, and all the other ones that we've had, like Sue Kobe gave us a talk last year, and we've had some just great speakers near you. Anyway, we won't go into all those people, but thanks to them as well for dedicating their time to this series. And I guess the other thing is we want to say is, if you do find some value here as bee breeders, and you want to kind of move this along yourselves, you can donate to our group, and those monies will go toward breeding projects, and we would report on those in the future, or become a member and participate in some of the decision making we make, and we'd love to have you join us. It's open to all breeders, not just BC breeders. Anyway, thanks again to everyone that's participated, both the questions and speaking, and have a great weekend. Oh, wait, I guess, yeah, I should also say who our next speaker is. So our next speaker is going to be Mary Lou Morin. She's from the University of Laval from Quebec City, Quebec. And she'll be speaking on hygienic behavior, VSH, recapping varroa levels, and NMR, or non-mite reproducing tests to select the best hives for breeding, what they've done to correlate those traits, and what they've found. So it could be just an absolutely super talk. That will be Wednesday on February 22nd of this year. And I hope you can all join us then as well.